Microbial metabolism is the process by which microorganisms take in nutrients, convert those to energy, and use that energy to fuel growth and other life processes. Metabolism is simply all the chemical reactions that occur in the cell. Catabolism is the breakdown of large molecules to smaller molecules. When we break bonds, we release energy, and that energy can be used for cellular activities. Catabolic reactions are said to be exergonic because they release energy. Anabolism is the building of new molecules. This is always going to require energy. Anabolic reactions are said to be endergonic. Oxidation-reduction reactions, or redox reactions, are the transfer of electrons between molecules. You'll remember that we need to think of electrons as little bits of energy, so wherever the electrons go, that's where the energy goes. When molecules lose electrons, we say they are oxidized, and when they gain electrons, we say they are reduced. Leo Gurr can help you remember this. Lose electrons oxidized, gain electrons reduced. Because electrons are little bits of energy, we don't allow them free in the cytoplasm. They could do a lot of damage. Electrons are usually associated with protons, and one electron plus one proton is a hydrogen atom. When chemical bonds are broken, electrons are released. Now, we have to do something with those electrons, and we may not have a reduction reaction going on nearby that's part of metabolism, so we need to have something that will hold those electrons. These molecules that will temporarily accept electrons are called electron carrier molecules. Not only will these molecules temporarily accept electrons, they will also easily give them up should electrons be needed in an oxidation reaction. There are three basic electron carrier molecules. Nicotinamide adenine dinucleotide is known as NAD when it's in its oxidized form, and NADH in its reduced form, the H indicating that it's picked up an electron. Nicotinamide adenine dinucleotide phosphate is NADP in its oxidized form, NADPH in its reduced form. This particular carrier molecule is used almost exclusively in plants. Flavin adenine dinucleotide, or FAD in its oxidized form, is known as FADH2 in its reduced form. Eventually, electrons have to land someplace where they won't be given up easily. We call this molecule the final electron acceptor. Catabolism is going to release energy, and energy needs to be managed. We can release a lot of energy with one catabolic reaction. Some of that will be used immediately, but some of it needs to be saved and used later. ATP is our energy storage molecule. We can store that chemical energy from the catabolic reaction in a high energy phosphate bond. We say that ADP is phosphorylated to ATP. Phosphorylation is simply the process of adding a phosphate to a molecule. So when we add phosphate to ADP, we have phosphorylated the ADP. We can phosphorylate ADP using three different mechanisms. We have substrate level phosphorylation, oxidative phosphorylation, and photophosphorylation. As we continue to talk about metabolism, we'll give examples of each of these. In this picture, we see an overview of metabolism. Nutrients come into the cell and they're broken down. We get energy, some of which is stored in ATP, and some of which is lost as heat. Energy can neither be created nor destroyed, simply change its form. Later on, some of this ATP energy can be used in anabolism, where we build larger molecules from smaller ones. These macromolecules can be cellular structures. They can be used for cellular processes like cell growth and division, or they can be converted to other macromolecules like carbohydrates and lipids, which may even go back into a catabolic reaction. Substrate level phosphorylation occurs when there is a phosphate attached to a substrate molecule. When we break the phosphate off, we get enough energy from that broken bond to add the phosphate to ADP. Oxidative phosphorylation involves a series of redox reactions. These redox reactions provide enough energy to pump hydrogen ions across a membrane. As hydrogen ions rush back across the membrane, it generates energy that converts ADP to ATP. This process of hydrogen ions rushing back across the membrane to generate energy is similar to generating electricity using turbines in a dam. Photophosphorylation occurs when energy is harvested from light, and that's the energy that's used to phosphorylate ATP. Enzymes are an important piece of metabolism. Enzymes are catalysts, and they are always proteins. We can tell we're dealing with an enzyme because it ends in ASE. Catalysts simply speed up chemical reactions. Every chemical reaction has a little startup energy that's required. We call this the activation energy. Enzymes lower the amount of startup energy any reaction requires so that the reaction starts more rapidly. Many protein enzymes are complete in and of themselves, but many have protein and non-protein components. In this case, the protein component is called the apoenzyme. Apoenzymes are not active unless they're bound to a non-protein cofactor, usually an ion, and or an organic coenzyme. Here you see in blue the apoenzyme, the protein portion. This particular enzyme, in order to become a holoenzyme, an entire enzyme, needs an inorganic cofactor as well as an organic coenzyme. When these two things are in place, the enzyme now has an active site. This is the area where the substrate will bind. For an enzyme to work, the enzyme must fit its substrate. This means that there is enzyme substrate specificity. Since enzymes are proteins, and we're always talking about how protein structure is important for protein activity, we have to have that protein molecule fitting another molecule. Here is another example of that. The protein enzyme has to recognize the substrate. The part of the enzyme that recognizes the substrate, the functional site of the enzyme, is called the active site. For enzyme activity to occur, the enzyme has to associate with its substrate. That is, the active site has to fit the substrate. The enzyme and substrate bind, so we have an enzyme-substrate complex, and then the substrate is altered. Bonds can either be broken or bonds can be formed. The enzyme will then dissociate from the new molecule or new molecules and go on to work with another substrate. So here we see a substrate fitting into its enzyme, and we have the enzyme-substrate complex. This is going to work only because proteins have that specific shape. Here we have a substrate. In this case, it's fructose 1,6-bisphosphate. It fits in its enzyme, and this particular enzyme is going to split this into two separate molecules. Once this happens, the two separate molecules are released. The enzyme can go on and react with another molecule. Most of these reactions are reversible. These two molecules could just as easily fit in this enzyme and be joined to become the larger molecule and then be released, and the enzyme can go on to put two more molecules together. Enzymatic reactions can be affected by environmental conditions. One of these conditions is temperature. Each enzyme has an optimum temperature at which it functions. If the temperature is less or greater than the optimum, then the reaction rate will be slower. At very high temperatures, because enzymes are proteins and high temperatures will denature protein, the enzyme will be destroyed. This means that the cell will permanently lose that enzyme. pH is another factor that can affect the rate of enzymatic reactions. Again, each enzyme has an optimum pH. Extreme pHs also denature enzymes. And the third thing that affects the rate of enzymatic reactions is the amount of enzyme and the amount of substrate available to react. If there's quite a bit of substrate, then every enzyme will be busy breaking down substrate. You'll have more reactivity. Once all the enzyme is bound to the substrate, however, you'll have your maximum amount of activity. If there's not enough enzyme concentration, then the enzyme will be very slow to break down the substrate. Enzyme inhibition can occur when substances block the enzyme active site. This is a way of inactivating an enzyme that does not denature it. So we don't destroy the enzyme, we simply inactivate it temporarily. Competitive inhibitors fit the enzyme's active site so the substrate can't bind. The inhibitor, however, is not changed by the enzyme, it simply blocks the active site. Some competitive inhibitors bind permanently, others bind reversibly. Sulfonamides are a type of drug that show irreversible competitive inhibition. Sulfonamides will bind to an enzyme that's required in the folic acid production pathway. Since enzymes need folic acid to survive, if we shut down this pathway, the organism will die. Non-competitive inhibitors bind to a site that's not the active site. This site is known as an allosteric site. When the non-competitive inhibitor binds, it alters the shape of the active site, so the active site will no longer fit the substrate. Now here is a competitive inhibitor. It sits in the active site, and the substrate can't bind. A reversible competitive inhibitor sits and will release, and then substrate can bind again once the inhibitor is gone. This is an example of a non-competitive inhibitor. Here, the allosteric site, when something binds to it, it forces the active site to distort. Now it will no longer affect its substrate. Again, this can be reversible or irreversible. Feedback inhibition is a way that cells can control enzyme action. Every enzymatic reaction requires energy. If cells really do not need a pathway to run, they need to shut it down, but they may not want to shut it down permanently, just temporarily. A good way to do this is with a negative feedback or end product feedback reaction. Metabolic reactions are a series of reactions. We usually give you the starting point and the ending point like it's just one straight reaction, but usually there are several steps to get from the beginning to the end of something. Enzymes are required for each step of that pathway. If we have an end product that can feed back to the first enzyme in the pathway and act to inhibit it, we can shut down the production of the end product. We shut down the entire pathway. When the end product has been consumed by the cell, it will even be consumed from that first enzyme. The pathway can resume again. This allows the cell to conserve energy but maintain an active supply of whatever the end product molecule is. This substrate eventually turns into this end product, but it is a three-step process. Enzyme 1 acts, you get this molecule reacts with enzyme 2, and you get this molecule, and this molecule reacts with enzyme 3 to give you this molecule. If we have plenty of this molecule in the cell, this molecule feeds back to enzyme 1, binds to the allosteric site, and alters the active site so that we can't act on this substrate anymore. As our end product molecule is consumed in the cell, it will even be pulled off of the enzyme, making the enzyme active again, and this pathway will operate again so that we can make the end product. Glucose is the primary molecule for ATP production in the cell. Cellular respiration may be either aerobic or anaerobic, and also fermentation is a pathway for utilizing glucose. All three of these reactions, aerobic and anaerobic cellular respiration and fermentation, start with the same step, glycolysis. In glycolysis, we take the molecule glucose 6-phosphate, and we need that startup energy of 2 ATP. And this is a 6-carbon molecule. We're going to break that in half. Glycolysis is lysing glucose. That's literally what we do. We break it in half and we get 2 pyruvate molecules, each containing 3 carbons. In the process, we make 4 ATP, and because we have broken bonds and released electrons, we have to reduce some NAD to NADH2. We get 2 of those molecules. Oxygen is not required in this step. And this is an example of substrate level phosphorylation. We take the phosphate off the glucose 6-phosphate. That provides the energy to put phosphates on ATP. Cellular respiration has an intermediate step now. We have to convert the 3-carbon pyruvate to a 2-carbon molecule known as acetyl-CoA. Again, we're breaking bonds. We're breaking a carbon off of the pyruvate, so we get some carbon dioxide. We liberate some electrons, so we reduce some more NAD. The acetyl-CoA is what goes into the Krebs cycle, also known as the tricarboxylic acid cycle, or TCA. Acetyl-CoA will join with a 4-carbon molecule called oxaloacetic acid to make the 6-carbon molecule citric acid. What happens then is a cycle of reactions where we break off some carbons, we rearrange carbons, and we get back to oxaloacetic acid again and just start the process over. It is literally a cycle of repeating reactions. As a result, we rearrange and remove some carbon atoms. We get some more carbon dioxide released. Because we have broken bonds and liberated electrons, we need to reduce some of these electron carrier molecules. We use NAD again, but we also use FAD, and we get 2 more ATP out of this deal. So to look at this reaction, there's the acetyl-CoA going in, the 4-carbon oxaloacetic acid, making the 6-carbon citric acid, and all we do, we occasionally break off a carbon, we liberate electrons, we break off a carbon, we liberate electrons, here's where we make some ATP, and we just go back around and we come up with this 4-carbon molecule again that will accept another acetyl-CoA. So far, we have not made a whole lot of energy out of our glucose molecule. Out of glycolysis, we got 2 ATP net. It took 2 to start and we got 4, so that's only 2 net. But we reduced some NAD to NADH, which means there's some electrons in this molecule, some energy in this molecule. In the acetyl-CoA conversion, we got a couple of CO2, but we got a couple of more of these energy holding molecules, these NADH. Out of the Krebs cycle, we get 2 ATP, we get 4 carbon dioxide, but again, we get some more of these electron holding molecules. 4 ATP is not a whole lot of energy to get out of a glucose molecule, but we do have NADH and we do have FADH2. These molecules are holding electrons, and electrons are energy. So we need to harvest that energy, and to do this, we're going to use redox reactions. This is where we need the electron transport system. The electron transport system is simply a system of electron carrier molecules. Electrons are transferred from carrier molecule to carrier molecule, and these carrier molecules are bound to the membrane. In a eukaryotic cell, they're found on the inner membrane of the mitochondria. In the prokaryotic cell, they're bound to the cell membrane. While the process is basically the same in both eukaryotic and prokaryotic cells, there are some different transfer molecules in the 2 cell types. As electrons are transferred from carrier molecule to carrier molecule, a little energy is lost. Not a lot, just a little bit. Just enough energy to pump a hydrogen ion across a membrane. The electrons are finally transferred to a final electron acceptor, that is, to a molecule that is not easily oxidized. Then we can produce ATP by the process known as chemiosmosis. We're going to create a flow of hydrogen ions, much like water turning a turbine at a dam. In the electron transport system, we simply put the NADH and the NAD in. We oxidize, reduce, oxidize, reduce, oxidize, reduce. Every once in a while we get enough energy to pump some hydrogen ions across that membrane. Eventually, the electron ends up on its final electron acceptor molecule. Chemiosmosis is how we're going to capture that energy that's lost during the electron transfer. We use that energy to pump hydrogen ions across the membrane. The membrane is impermeable to hydrogen ions. That's why we had to have a pump. On one side of the membrane, we're going to build up a lot of positive ions. The other side will be positive ion deficient or more negative. So we're going to have an electrical gradient that will develop. There are few protein channels that exist in the membrane that will allow hydrogen ions to come back across the membrane. As hydrogen ions flow, they're going to generate energy to produce ATP. The electron transport system will generate 34 ATP. Now we have made some ATP from that glucose molecule. So as we look at chemiosmosis, here are some of those key electron transport molecules that pump the hydrogen ion across the membrane. We make it more positive on one side of the membrane and more negative on the other. These few channels that allow the hydrogen ions to come back through are associated with an enzyme called ATP synthase. This is going to take ADP and phosphate. The hydrogen ions flowing through will give us the energy and will make ATP. So we have a strong electrical gradient here, positive this side, negative this side. When we open up this gate and let these hydrogens through, they flow very, very quickly, and that is what gives us the energy to make ATP. This is an example of oxidative phosphorylation. Those final electron acceptors are going to vary depending upon the organism. Organisms that rely on aerobic respiration will have oxygen as their final electron acceptor. Because the electrons have been carried as hydrogen ions, will make water, H2O. These organisms are said to be aerobic. In anaerobic respiration, there is an inorganic molecule that's the final electron acceptor. Something like sulfate is reduced to hydrogen sulfide. Nitrate may be reduced to nitrite or even to nitrogen gas. These organisms are known as anaerobic organisms. There are some alternates to glycolysis. The pentose phosphate pathway takes a 5-carbon sugar. And early in the glycolysis process, there are some 5-carbon sugars formed, and these 5-carbon sugars can, in certain organisms, become part of the pentose phosphate pathway. This pathway is not really used to make ATP, but it's used more to make precursor molecules that are used in anabolism, making things like nucleotides and amino acids. The Entner-Deuteroff pathway is an alternate route to make pyruvate. Instead of going through glycolysis, it goes through a slightly different pathway. The pyruvate then turns into acetyl-CoA and feeds into the Krebs cycle. Fermentation starts with glycolysis but doesn't go through the respiration pathways. Rather, it goes to fermentation. In this case, organic molecules are the final electron acceptor. Again, this is not so much used to generate energy because you get very little ATP out of fermentation, but it does help oxidize NADH and FADH2 back to NAD and FADH. Oxygen is not required for fermentation. You will remember Pasteur's germ theory of fermentation. He said that different organisms would give you different fermentation end products, and that's what we see here. Propionibacterium gives you Swiss cheese. Things like Aspergillus and Lactobacillus and Streptococcus can give you cheddar cheese and yogurt and soy sauce. The yeast Saccharomyces can give you carbon dioxide and ethanol. And Clostridium gives you acetone and isopropanol, rubbing alcohol. What happens as these products are made is we oxidize NADH back to NAD, and this is how we generate NAD to go back through respiration. So to recap the overall processes here, fermentation makes 2 ATP. We only get the 2 ATP out of glycolysis. Organic molecules are the final electron acceptor, and this is primarily to generate NAD so that we can go through the other processes here. In aerobic respiration, we get about 36 ATP. Oxygen is your final electron acceptor, so you get water, and we go through glycolysis, the conversion to acetyl-CoA, the Krebs cycle, and the electron transport chain. In anaerobic respiration, we get more than 2 ATP. We get more than what we would get out of glycolysis, but we don't quite get as many as we get out of aerobic respiration. The electron transport chain is not quite as efficient with anaerobic respiration. We still get a good many ATP. The difference between aerobic respiration and anaerobic respiration is the final electron acceptor. This is going to be an inorganic molecule. Other catabolic pathways include the way we break down lipids. Lipids, you'll remember, are glycerol and 3 fatty acids. Well, glycerol is a 3-carbon molecule, so it's broken off and becomes pyruvate, converts to acetyl-CoA, and goes through the cycle. The fatty acids can be broken down 2 carbons at a time. They can become acetyl-CoA and enter the Krebs cycle that way. All of the molecules end up entering the Krebs cycle fairly early on. Proteins are not a particularly good source of energy. The proteins have to be broken down to amino acids, and then we have to break the amine group off. This is a process called deamination. We either make ammonia, which is part of the nitrogenous waste products of an organism, or we'll recycle the amine group if we build new proteins. The remaining part of the molecule may enter the Krebs cycle, but it enters pretty late in the deal, so a lot of proteins don't make very much energy at all. The other piece of metabolism is anabolism. One form of anabolism is photosynthesis. Photosynthesis is going to harvest light energy. Cells that do photosynthesis have chloroplasts and chlorophyll in them because this is how we're going to capture this light energy. We'll use carbon dioxide and water and photophosphorylation to make ATP, and we'll also make carbohydrates. Plants store their excess carbohydrates as starch. All cells will undergo biosynthesis. We have to form glucose, proteins, lipids, and nucleotides all the time to build new molecules. Most molecules in a cell are what we call amphibolic molecules. That is, they can be used in catabolic reactions or in anabolic reactions. As we're breaking molecules down, we sometimes make the building blocks that we need for anabolism. Basically, anabolism is just the reversal of catabolism, and most of these reactions are two-way reactions. Gluconeogenesis is the process of making glucose from some molecule that is not a carbohydrate. This rather busy picture shows you the interaction of all of those amphibolic molecules. We have proteins, nucleic acids, polysaccharides, and lipids are major macromolecules. We need to make these. We can also use these as nutrients. If polysaccharides are broken down, they can go into glycolysis, the Krebs cycle, and we can make ATP. But we can also go the other way. We can take some of these molecules and build polysaccharides if that's what we need. Proteins have to be broken down to amino acids. We have to deaminate, or we can save that ammonia and add it to some other molecules and make amino acids. As we break the amino acids down, notice that they can go into different areas of the glucose catabolic pathway, or we can pull molecules out of those pathways to make new proteins. The same is true for nucleotides. They can be broken down and feed in, or we can take pieces out to make nucleotides. Lipids can be broken down to feed into glucose catabolism pathway, or we can pull molecules out and make new lipids. All of these are examples of amphibolic molecules.